Okay, I guess we'll kick it off. Right, it's almost 5 by 3. So welcome to our panel discussion, which is focused on characterization and purification of peptides and oligos. So as we've heard today and probably for the last few years, those two modalities are definitely growing in importance and taking a lot of attention. And as with any new modalities, I think the challenges remain or pop up. And today what we decided to focus on is predominantly on those key areas that I guess most challenging from analytical standpoint as well as purification. But before we go into our discussion, well, first of all, we want to keep it totally informal and open for comments, questions during our panel, so it's not supposed to be just us talking. But let us start first by introducing ourselves, and then we can move on into our questions. But again, we will first talk about analytical challenges and in characterization. Then we'll talk about purification strategies and scale-up. I threw in there some fermentation for peptide production for those of you who might be still interested in recombinant peptide production. And then if time allows, we will talk about some innovation and future outlook and see what's happening in that space. So with that, let's go. Who wants to go first?
I have no problem going first. And thank you, Masha, for moderating today's session. My name is Art Kasson. I work for a company called YMC America. YMC America is a subsidiary of YMC Corporate and based out of Japan. Here in the U.S., we are based in Devens, Massachusetts, about 45 minutes northwest of Boston. I cover sales in the Northeast U.S., but I have experience as a lab manager and a bench chemist. I used to do mass spec up at a couple universities. I had one up in Ithaca, New York, and then one down in the Houston area of Texas. In fact, we were just up here talking about the times we lived down south. But yeah, today we've seen a lot of things happening in the field of peptides. YMC as a company is probably most known for being a preparative chromatography company. So while development is being done on a lot of different modalities, we're seeing a lot of companies contacting us about larger size resins and more robust columns and larger skids that can handle the sort of capacity that's needed by a lot of the big companies that are manufacturing and producing peptides. So I'm excited to be here today, and thank you all for coming.
Sure, I'll go next. So Frank Tagliaferri, I'm CSO of Pace Life Sciences. Pace is probably not the best known CRDMO, but we've been around 20 years, though we've expanded a lot over the last 3 years. So we have 9 development, testing, and manufacturing sites around the country. And so we focus a lot in terms of this panel on oligonucleotides and peptide analytical characterization and development and getting them into the clinic. So.
Okay. So I'm Masha Kononov. I'm with Ingenza, which probably also not a very familiar name in the space. We're actually we've also been around for over 20 years, and our specialty in a nutshell is process development, but we are mainly focused in biologics space, but we have been working a lot in the last few years, and particularly with peptides, but more on the recombinant production, I would say. And we do a lot of characterization work as well as we have high-end analytical tools. So we've seen a lot of challenges with peptides, in particular in the characterization and impurity analysis. So that's in a nutshell about us and me personally.
Sure. And hi, everyone. I'm Stephen Calciano. I work at Waters Corporation within the Bioseparations division. Waters is a manufacturer of liquid chromatography, mass spectrometry, and a whole host of other characterization tools, including particle analysis now, and multi-angle light scattering. So there's a variety of tools that we use to help and assist in characterization of peptides, oligos, and other biomolecules. And yeah, I'm excited to learn more about the panel and the audience here. So I'm looking forward to a good session. Thank you very much.
Excellent. So as I mentioned, we have a little plan how we want to run this. So the first thing we want to focus is analytical challenges and characterization. And I think all of us agree that both peptides and oligos face similar hurdles, such as detecting trace impurities, confirming structural identity, and ensuring reproducibility, and then, of course, impurities. So would you agree with that? And maybe share your views and your experiences, and maybe something, other hurdles and bottlenecks that you're saying. And the same question is to audience. If we're not touching on something that you are experiencing, we're happy to hear from you and maybe address those points, questions as well. Who wants to go first?
I can kick it off. Yeah, no, I think it's, as the modalities have advanced, it's particularly around peptides, I think some of the newer products that are coming to market are seeing more modifications to the peptides that are, challenging the existing methods that are used. I think the impurity profiling methods I've seen evolve a little bit over time. Traditionally, most of our customers were using C18 phases, and I think as we've started to experience or encounter more fatty acid modified peptide backbones, we've had to sort of take a fresh look at the impurity methods and phases we're using to resolve out some of those impurities that are problematic when using just a typical C18 phase. So I think that's something I've observed looking at different ligands like phenylhexyl ligands. Has been a way that we could sort of, overcome those challenges. But yeah, I think the complexity of the modality and how they've sort of evolved to extend like PK profiles and, address some of those degradation concerns have sort of challenged how those typical impurity methods were originally, designed, and certainly makes for interesting challenges, I think, from a chromatographic perspective.
Sure. Yeah, I think where I come from with this is it's the analytical challenges are there, but the real key is actually to convert those to a QC setting. So in the early development labs, we'll get a new molecule in, we'll hit it with a NMR or an Orbitrap, and so we'll characterize the full molecule and get the impurity profiles, and we'll throw it on the SecMols or an AF4 or whatever we need to do. But the key is how do you transfer that to the QC lab where we're doing the release on that, and so that's the real key, to kind of pick key parameters off these kind of fancy, expensive instruments that aren't in the QC lab and look at those to help with ID testing or impurity testing and things like that. So, to go with something like water, so to convert everything to a chromatography method would be great. We do that a lot in the RNA space now, right? So I don't have to do CE anymore, so I can convert it to a reverse phase method that we can do in a QC lab. So that's the challenge that we see.
Yeah. So sort of bridging that gap between what you can do in extended characterization and then bring that back to something that's reliable.
It takes a lot of CapEx requests to get a couple of Orbitraps. But,
Definitely.
It takes a couple CapEx requests to scale it up to a prep chromatography instrument as well. But at least from the analytical challenges we've seen, I think in terms of impurity detection, having LC mass spec ultra sensitive detection is going to be critical. We look at orthogonal HPLC and different modes using C18, and Steve's correct. As scientists are starting to move away from some of these fatty acid backbones, we need to be prepared for the future, like especially as we're developing towards a scale up, and we'll talk a little bit more about that. I feel like I know what works for our own customers at YMC, but I don't always get the full picture because I'm right there in the middle, trying to help them scale up to a prep chromatography system. So we do have some interesting things going on, especially in the peptide world. But remains to be seen what will happen next.
Yeah, and in our world, I mean, we are seeing the same challenges, but we are a development company. That's our main focus. We do process development. So for us, characterization of the molecule is the key, and to have high-end analytical tools is critical, because some of those structures that we are seeing are quite intricate, and a lot of work that we do is actually when there's a multi-component aspect, and so in some instances it's very hard to distinguish between not only talking about impurities but the molecules themselves, the peptides. So we actually have Q2MS that served us quite well to do high-level characterization, not only of impurities but the molecules themselves, the peptides. Excellent. Any comments or questions from the audience? Any challenges that we have not touched base upon? Okay.
I know, at least from the reproducibility side of things, this is always something that whether you're doing the first stages of development, having a column, having a system that's reproducible from day to day and minute to minute and hour to hour, that becomes important, and it really sets, I know, at least in my mind, when I'm talking to a customer about reproducibility, I want them to be able to not just have to worry about reserving a batch of analytical columns for the next 3 years because they're all from the same lot. I want them to feel confident enough that if they are buying one of our Tri-Art C18 columns, as an example, that they could be buying the same thing 2 or 3 years from now and getting a similar result and not having to worry about that. And that would obviously be coming from a different lot. It's not something that they should need to reserve. And I know when we're talking about chromatography from a standpoint of oligonucleotides and peptides, we're mostly focused on reverse phase, or at least from my customer base, ion exchange. And those are the two modes that we use. And there are limitations, at least on the ion exchange end, especially with our non-porous. The non-porous ion exchange resins that we have only go up to 6 micron particle size. So then our customers are forced to use a porous media. And those are the kinds of challenges that we're trying to get around right now. And I know that reproducibility is an issue, because it's one that I talk about quite often with our customers.
Yeah, I'm glad you brought up the reproducibility, because for us in one particular project that was also very important matter. And actually with this Q2MS, we had a great batch-to-batch reproducibility with results, and we're able to see a really tight, precise differentiation between those mixed peptides.
You got a question?
Quick clarification, if you don't mind. So I'm trying to get a feel of the scope in terms of when you're characterizing customers in the research stage, let's say, to Frank's comment, when it becomes a pragmatic QC step. Are you talking essentially, can you get it down to where you really just need one instrument or two instruments at the QC level, in terms of you're identifying what's the important thing to be able to characterize, and then selecting that? Even though you might not be seeing everything that's there?
Yeah, I think that's it. To be honest, it's not really the ID. You know, we can do a number of different ways, so you can get that down. But to do something like prove the the 2D structure, the 3D structure. What can you do without having to do an NMR or something like that? So look for an FTIR band or something that you can identify, saying that the peptide's in the correct conformation. Things like that are what we're trying to do. And the same thing with mass spec. You say, yeah, we can do a top-down full approach and sequence everything, but is there just a piece of information I can get on a triple quad or a single quad that they might have in a QC lab in order to kind of pick something out, so they don't have to kind of deconvolute a whole set of data.
Yeah, I think the idea is that if you can, through extended characterization, get the full scope of the compound molecule that you're interested in, and then as you proceed through the development, you understand more about what property of the molecule is most important to maintain the efficacy, the safety, and those become the attributes that are most critical for you to track throughout the development, manufacturing, and ultimately the attribute you select to test during QC and release of that compound. Not every attribute that you can measure should or could be measured as you move throughout that drug development and ultimately that release process. And I think that's why a lot of companies that have established techniques like CD or chromatography, that a lot of infrastructure is built around having those deployed in those sort of later stage settings for compliance and things like that allow you to sort of funnel in and develop methods that can characterize the things that are most critical for you to be able to support and prove safety and efficacy of those compounds.
A little bit of a follow-up, but are you finding that companies already have the infrastructure in place in QC, or are they going to have to invest in the instruments?
Yeah, I'm kind of only work with my own company, so we've made sure we've had that in place. But I guess like an example would be something like an oligonucleotide, where when we first get it, we're going to do a full impurity profile, look at the N-1, N+1, and do it the nth degree. We will try and convert that to just an HPLC trace where we can identify the impurities we're in charge. So most labs will have that. I think, like I said, it's the mass spec is the tough one to figure out what kind of instruments people have in their various QC labs and how well they run them, to be honest.
Yeah, and I think it's not always the case that you end up with high-resolution mass spectrometry in QZ. It could be that something as simple as mass detection is enough for you to get the job done and prove identity once you have your process well established. And then there's different levels and flavors of the instrumentation that some may be more deployable in QC. They're a bit more easy to keep operational. They may not be as finicky, and I think that all comes down to design as well and having the right tool for the right job at the right stage. So I think it's all about having the right technique to serve and answer the question at that stage. But I think in terms of CapEx investment, it comes down to, is there a new feature of the compound that's important enough to measure in a QC setting that you don't have capability for, and that's going to drive investment. In our case, I think we've seen a lot of examples of oligonucleotide analysis where you just can't get the level of resolution that you need with maybe HPLC, and you have to invest to go to smaller particles, and that will then force your hand to invest in higher pressure capable systems. So it's just is there a need that you have to address with your control strategy, and then that may force your hand in terms of CapEx.
Yeah, I guess just as an example, a company like Ionis, they've really developed a single quad analytical method to characterize their oligos, and that's a nice system because they can put it in labs and it's low cap and they can run it that way.
Yes, exactly. It's a very we see that method everywhere.
Yeah.
Yeah, and in terms of the money that's being allocated, I cover a lot of territory. I cover Northeast for YMC, and that means I'm traveling from Maine down to Maryland over to Ohio. As part of what I do in my day-to-day, I also visit with a lot of universities. And one of the trends that I've noticed in a lot of these chemistry departments where you used to see individual researchers or individual professors, they would come in, they would get some startup money, and then they would get whatever they wanted from their department. And it didn't matter if you went down a hallway at, say, Princeton, and there were 10 competing labs going down the hallway, and they all had the same pieces of equipment in each individual lab. Now, a lot of those same universities are consolidating that equipment into like core facilities that are being run within that department. Not only does that foster some more collaboration at these earliest of stages, but I think it ultimately saves these universities money. For an equipment manufacturer, it might not always be the best thing just to have your equipment as part of a core facility, because one of the drawbacks to that is then people have to schedule time on an instrument, and if there's more of a need, maybe they can get one or two more pieces of the same equipment. But a lot of times, that turns into the bottleneck.
All right. Any other comments, questions? All right, so we can move on to purification strategies and scale up, and I think that's probably more for Art than anyone else on this panel, just because we are probably more on analytical and characterization. But the question would be, so what are the often bottlenecks that you are experiencing when you're moving in production from smaller scale to larger scale, and what are the key challenges there?
Yeah, I mean, there's a lot of challenges in this area, and it really depends on what's being scaled up. But we talked about this a lot yesterday in our workshop. But one of the challenges with the oligonucleotides is certainly the range of pH buffers that are being used, and then also looking at the shape, and there's a whole bunch of other factors that can really influence how difficult it might be to separate and purify this out. Our workshop yesterday was on why continuous chromatography is a good option for that scale up in some instances, and our application scientists and our group from YMCA India helped with that yesterday, and we really came up with some unique situations of where these can be challenging. Scale up is not always linear. I think that's that's the key point, and you hope that it is, but there's always something to screw things up, and it might be something like someone needs they're scaling up from a 4.5 millimeter column to a 200 millimeter column, but ideally they should be using something like a 218 millimeter column, which there aren't many vendors who make that. So you can calculate certain things out, but that's just one challenge of trying to scale things up from even the pilot stage to then production stage. And really that's, once we get up to like prep chromatography at like 30 or 50 mils per minute, that's kind of where YMC starts with how we think of chromatography. But there's been a whole bunch of development work that's been done before then, and a lot of that, I'd say 90% of that is done on the water system. I mean, every lab I go into, I has everything from an alliance to an acuity. So I know that we have to be able to scale up from a system that we don't manufacture to a mid-range intermediate system like a benchtop prep chromatography system to then a production chromatography system. And there are challenges with each of those steps, and it depends on how many of the intermediate steps that someone actually wants to put in there before they commit the millions or tens of millions of dollars that are going to be required to do the full scale up to that 80 liter per minute skid with huge pumps that goes all the way up to the ceiling. And that's just the tip of the iceberg of where these challenges begin. I could probably spend the rest of our time talking today about those challenges. I am happy. I won't do that, I promise. But I will, if anyone has questions about that or wants to talk about that, I could be available after we're done here.
Actually, so the last talk, we spent a long time saying that the GLP-1s are going to use the world supply of acetonitrile, and you're using ion exchange. So, are there scale-up methods that are going to be?
Well, that's what one of the major challenges we're having right now is, at least for YMC, the largest non-porous ion exchange resin we have only goes up to 6 micron particle size. You're really limited in back pressure and ID on chromatography columns when the largest particle size is 6 micron. We also have porous ion exchange media, and our ion exchangers are strong versus, we have strong anion and strong cation exchangers. I think when you take the porous, our largest particle size is 75 micron. So those go up to a pretty normal size for what people use. I think 10, 20, 30, 70. There might be a 50 in there somewhere. So that's a challenge in itself, because the resolutions between the non-porous and the porous materials are so different. So that's a challenge right there, especially if we're, like I said, using up this supply of ACN. And then, one of the other issues is that a lot of times there are limitations on even putting larger particle for ion exchange into stainless steel. You run into all these different materials, different solvents, and depending on, you can coat steel in a lot of different coatings. We actually have a branded bioinert coating. I know Waters has something similar as well. Those are the kinds of coatings where, especially if you're dealing with really acidic environments, those might be required. But for ion exchange, a lot of people just use glass. The limitation there is now you're dealing with a low to medium pressure issue versus what you can usually achieve using stainless steel. The problem with stainless steel is, at least with ion exchange, it's tough to pack a wet gel into stainless steel and get the kind of reproducibility that customers want. Mm-hmm. So there's a lot of failure rates, and we're talking about QC. The QC on stainless steel packed ion exchange is, there might be a 10% failure rate, and so those have to be repacked, and that leads to higher cost per column. Mm-hmm. And so when you're looking at a column that's 150 by 30 millimeters, and it's only packed with a little bit of ion exchange resin, but it costs in the tens of thousands of dollars range, the question we'll get is why, and that's exactly why. It's better to pack it in glass and pack it yourself, and then it's not as expensive. But you're limited by pressure.
Sorry, it might be a more basic question, but help me out. When you're scaling up essentially surface area, right? So when you think scale up, you essentially multiplying the surface area with resin that you need, corresponding flow rate and things like that. But my question really is there anything that we should pay attention to, not to change temperatures, pressures, whatever the case? What really affects these molecules in terms of scale? I mean, what do you want to keep constant versus what are you more flexible in terms of change? In other words, you might be more willing to change flow rates, whatever the case is, but you might, and I'm guessing, I have no idea, or you might not be, you might definitely don't want to change your potentials or your temperatures or something like that. This is just a general question.
I know at least. Why don't I'll let someone else take a chance. So,
Yeah, I think it could depend on what the critical pairs are you're looking to separate and purify from. And if you have a method that's suited for analytical and you're looking to scale up, the purpose of the method may be different. Maybe you're looking for the best resolution of some of your impurities from your main peak for characterization purposes. And from that perspective, scaling up would involve you maintaining really tight control over everything like temperature, flow rate, because those are going to be critical to establishing the same exact separation. For purification, it could be that you are looking to just take the high purity cut from a main peak, and you can sacrifice a little bit to improve yield or to improve speed. So I think there's all of those. I think they call it the triangle, right? It's yield, it's speed, it's cost, and there's all of those things are in balance. And it just depends on the requirements of the assay. So if your purity requirement's 99.5, you know that you have to really push for purity, and that those levers can't be adjusted like speed or other things like that. And you may be sort of forced into a position where you're going for ultimate purity. So it's not always the case that things can't change, but just because a method is made for something like analytical characterization, let's say it's using an LCMS compatible buffer, but going to semi-prep or prep scale, that's not a concern, right? You're not going to mass spec. So you can alter your mobile phase additives to drive toward yield because you're not necessarily going to take effluent and go into a mass spec. So the parameters are always adjustable. It just depends what's the critical thing that you're going for. If it's purity, if it's speed, and then maybe my other kind of.
I was going to say my general rule of thumb: peptides don't like to change pH much. And
Yeah,
oligos don't
solubility.
Oligos, if you're annealing thing, you don't really like to mess with heat too much. So
Right.
Those are the two things
Yes,
I worry
I agree.
about. Yeah.
I think to add peptides for scaling up, we see like the volumes of quantities to be purified, so that's the scale up. But mostly the constant, like stationary phase should be the same.
Mm-hmm.
Scaling down from analytical to process phase. So,
Mm-hmm.
unless like you fix it the stationary phase, remaining as you said like fluoride, fluorides, the idea of the scale up, so linear scale up, so that can be as a leading gradient scale up. But the quantities, when you take the analytical, so the in ml and 5 liters, but taking the liters, 40,000 liters in the scale up to handle this and handling the 1 liter, so it's totally the
Mm-hmm.
so those are some critical parameters to be.
Yeah.
Then the storage of a peptide after purification, how many hours you store it? So maybe for 10 hours to distill it out of the lyophilized, it takes volume base. So that's, I think, the key parameters to
Yeah.
check out.
Yeah, and one cautionary tale that I could probably send out to everyone in the room that's doing HPLC is to make sure that whatever the column and whatever the stationary phase is that you're starting out with at the mass spec level, that it has the capability to, once you get it past an analytical size column to a semi-prep to a prep column in terms of scale up, that stationary phase is available at the larger sizes that you need it, because not only do you, when we talk about reproducibility, do you want to be able to make sure that an analytical column from lot to lot is behaving the same. You want to make sure that when you're doing UPLC to HPLC to prep HPLC, that has reproducibility as well, and making sure that you have a stationary phase that's available in 3, 5, 7, 10, 20, 50 micron particle sizes becomes important, because once you get it to that larger production stage, you need a larger larger size resin. So I think that's important to understand at the very beginning before you spend millions of dollars on developing something that can't be scaled up.
Yeah. Any other comments? No, we are a biology company, so we do a lot of peptide engineering using live systems, microbes. So of course, in our case, in our hands, pH and all the other parameters are critical. Temperature as well, actually, to make those at scale. but I don't think we're going to talk about fermentation or anything like that in this panel, because I just heard from previous panel that everyone believes that.
They didn't have
But,
any
but
interest.
yeah, no, that everyone believes that going back to biotechnology or recombinant production of peptides is not something that peptide industry is going to be going back to. But we, in our, in our company, we do have a lot of projects and interest from our clients to use live systems to make more complex, I guess, more higher molecular weight peptides. And again, it depends on what audience you're talking about, whether peptide is a small molecule or biologics. We call them mini proteins, but in a lot of cases, they resemble the same features as the smaller peptides and have similar challenges. There's anything else we want to touch? I think maybe advanced tools such as machine learning, AI, and application of those in any of your companies. Is that something that industry is moving forward with? I just came from another conference a couple of weeks ago, bioprocessing, and it was all around AI and ML, particularly analytical development, and how powerful those tools would be or are for data processing, for even designing of experiments and things like that. So any comments, thoughts on that? And it's also open to audience, I
don't know. In terms of AI, the thing we're touching on just a little is basically using it to predict peptide structure from less information that you usually have. So not having to blast the database and things like that, and kind of go into some deeper learning and things. It's not our expertise. We're tapping into some other people there. Anything that helps speed the processing and kind of simplifies it would be good. And then I think your other point was just automation. So I think automation, again, kind of in terms of the sample prep or some platform methods. We work on oligos. They're 20 mers. They're all basically the same. But if we can come up with some platform methods and automate the analysis and the sample prep of those molecules, would be nice. And to be honest, GLP-1s are the same thing, right? You have 30 amino acid peptides that they're a little different, but to be honest, we probably have a dozen of them in the lab. So we should be able to take advantage of the similarities to get some speed here.
I think the other area that I've seen new technology play a role is in the manufacturing of tirzepatide from Lilly. They're using continuous manufacturing, and they've actually started to, or they have introduced inline impurity and intermediate characterization. So they use that to basically take a multiple batch stream process into a continuous manufacturing process that was a hybrid solid phase, liquid phase, to basically bring all these different intermediates in line together using, it's called a patrol system, which is essentially an LC that plugs into your manufacturing process. And I thought that was a really interesting case of seeing inline analytics support process flow control and their ability to support and scale production very quickly. It was a unique case that I saw that I found particularly interesting. So. I think it's a nice example of how new technology can make something like a continuous process seem less daunting.
I'll be interested to see how this new 11 or 12 $billion facility that they're building out in Indiana turns out, because I think a lot of that's going to utilize a lot of these continuous manufacturing processes. And I don't know exactly how all of my customers are utilizing AI. I know that, at least from the perspective of sales, there's always people coming to us and saying, we can modernize our human resources department. They can make like I don't have to make as many phone calls a day if I utilize this tool. I was born in the 1970s. I'm Generation X, and I still have I still had trouble after COVID when I couldn't travel like 75% of the time, because that's what I was just used to it when I was on the road. So scaling that back to like 50% after COVID, I thought was like doing enough on Teams meetings was advanced enough for me. But I know these tools are going to be utilized over over the next decades. And I think there's good things that are going to come from them. There's bad things, and I think we have to be able to understand the difference, and that's going to be whatever industry we're in. The stuff's here to stay. On my way out here, I drove out on I-78, and all I could see were data centers and warehouses coming up, and it's the data center revolution right now. So I think there's no stopping it.
No, absolutely. No, in our experience, I mean, we are implementing a lot of machine learning algorithms to do discovery work. We actually do offer that as a service, looking for novel peptides and enzyme discovery. And of course, that first step for us is to do a lot of bioinformatics searches in databases that analyze, predict the structures and predict certain things, and then have a pool of much smaller number of peptides that we can then go and test using assays for activities. But we're also trying to implement a lot of AI, I guess, or bioinformatics tools in data analysis as well, because the more data you generate, the harder it is and more time consuming. So I think it's also adding efficiencies and staying competitive. So I think this is pretty much all what we wanted to discuss. Unless there's any questions or comments from the audience, we're gonna give you, no, we're not gonna give you any time back. It's exactly 3:45. Any comments? Thank you very much, everyone, for coming.
Thank you.